Let's get a few terms down before we get too far into things. We will talk about analyses, determinations, and measurements in class, but they are not all the exact same thing. An analysis is the actual process by which we use specific measurements to make a determination. We almost always have a process which requires us to measure a physical or chemical phenomenon that is not actually what we want to report a number for. For example, an analysis of blood glucose actually measures electrochemical potential from the oxidation of glucose to determine the glucose concentration. Try looking at the method attached to Brightspace. What is the actual analysis, determination, and measurement for that method? We also distinguish between techniques, methods, procedures, and protocols. Techniques are the broadest, and that's the majority of what you'd learn in this class or in instrumental chemistry. It's usually centered around the proper use of a specific piece of equipment, be it a burette for a titration or a microwave plasma atomic emission spectrophotometer for a concentration analysis. A method is an application of a technique for a particular type of analysis. For example, we might have different methods for looking at different metals or in different media. Samples of lead may require pre-treatment with hydrides, for example, while samples with sodium would not. Or we might even look at lead from different media, and that might be different, like we see for the methods lead in soil or lead in water or lead in blood in the graph. Methods are applied either as procedures, ways that the testing can be done, or protocols, which usually refers to ways testing must be done either by public policy or law. Many agencies may have different procedures or protocols that are published, including the APHA, which is the American Public Health Association, the ASTM, which is the American Society for Testing and Materials, and the EPA, the Environmental Protection Agency. So again, look at the EPA method 7196A, which is on Brightspace. What is the actual technique that's being used? What's the method? And then what's this procedure part of that? There are two main types of analysis, a total analysis and concentration techniques. In a total analysis, an entire sample is used for the determination. We may precipitate all the chloride out of solution to take a mass for a gravimetric analysis. So we take the entire solution, get all the chloride out, and we measure all of it. Or we may titrate all of the acid in the sample. Or the measurements may be as simple as all of the mass or all of the volume in a sample. These types of measurements generally require simple equipment, but more sample and generally more user skill, and also more time from that skilled user. On the other end of the spectrum are concentration techniques that generally require less time, less skilled users, but also require more specialized and also very expensive equipment. This includes spectroscopy, electrochemistry, and mass spectrometry. Pretty much if it's an electronic instrument besides a scale, it's a concentration technique. When we are selecting an analytical method, we need to consider what information we actually need from an analysis. I've given three scenarios that we might want to measure chromium in: industrial effluent, in waterways, and if we're just looking for chromium as a component in, say, like planing material. The accuracy, how close something is to the true value, is always going to be important for every measure. We measure that by percent error, just like we did in lab this past week. The precision, measured by standard deviation, is also really important, but to varying degrees. For example, if we're looking at the concentration of chromium-6 in industrial effluent, the concentrations are likely much higher than if we're looking at it in urban waterways. And so a measurement of 100 micromolars, plus or minus 10 micromolar, may actually be a reasonable level of precision for industrial effluent. But if we're trying to measure 3 micromolar chromium-6, that same plus or minus 10 is actually going to really be a problem. So how much we're looking at will determine how precise we need to be as well. We also need to consider the sensitivity of our method. The method 7196A that's on Brightspace uses a dye that changes color in the presence of chromium. How much the color changes will determine the sensitivity of the instrument. The graph on the right shows 4 different wavelengths at which a measurement could be taken.
It's hard to see, but the squares are the line that have the greatest slope. They correspond to 660 nanometers. That means any concentration of analyte will generate more signal at 660 nanometers than any other measured wavelength. This method is then the most sensitive at 660 nanometers and the least sensitive at 620 nanometers. The more sensitive a method is, the easier it is to measure low concentrations. It has a low detection limit. Generally, this is better. So why would anyone choose a different wavelength? Well, a lot of things might absorb at 660 nanometers, and if they happen to be in the same cuvette, you might choose a less sensitive wavelength because it might have less interference from other molecules. Or you might choose something a little less sensitive because it's safer to use. The dye structure that's shown on the left is arseniazo 3 that I used in graduate school for determining uranium concentrations. If I didn't need a really low limit of detection, if I didn't need to detect very small amounts of uranium, I might have actually used something else, something less arsenic-y. So which of these three scenarios, industrial effluent or urban waterways, or just if it's present in plating material, do you think probably needs the most sensitive detection? Speaking of interferences, ideally we don't have any, and we'd say that our method is specific because it has no interferences. If a method is not specific, we hope that we can at least be selective. That is, that we'd get more signal from one analyte than another. A common application of this is with DNA and protein measurements. Absorbance at 260 nanometers is selective for DNA and RNA over protein. You can see it on the graph on the left. But protein also gives a signal at 260 nanometers. Protein absorbance is at its maximum at 280 nanometers, but DNA and RNA also give significant signals. So it's not selective. But we can use both wavelengths together to figure out how much there is of each. Our two final considerations for a method are robustness and ruggedness. Robust experiments aren't prone to interferences. So a single wavelength determination of DNA would not be robust because we know that protein is a likely interference. A two wavelength determination would be robust. A rugged experiment does not change significantly in different experimental conditions. So in the experiment on the right, you see a thorium analysis is performed under three different strongly acidic conditions. Changing the acid doesn't significantly change the signal, and so we'd say that that method is rugged. We also have to consider scale of analysis. Some of this is determined by the mass of the sample we begin with. It's always easier to find low concentration materials when you have more material to work with. Methods all use some form of equipment. Simple equipment is generally inexpensive initially, but it will cost more time for a user. On the other hand, complex equipment is more expensive, but it's often faster and easier to operate. On the other hand, sometimes the best equipment can't tell us what we need. On the right, we have a microwave plasma atomic emission spectrophotometer, which is really good at determining element concentrations. What it can't tell, on the other hand, is what oxidation state something was. So if we're looking specifically for chromium-6, we would need another method, even though the microwave plasma would be a much better tool for measuring chromium itself in any oxidation state. So we might take a procedure off the shelf, or we might generate one on our own. But there are a lot of variables at play that will determine how reliable the data that we get will be. But they also determine how much person or money cost is required to get that data. And we'll always be bound by the sampling that's already been done. We can only measure the samples that are given. Two things that make it into many procedures to help with this are method blanks. A method blank would be like when we take a container and fill it at the sampling site with DI water and then put it through all the same steps. If our blank has something in it that we're looking for, that shows us that we're contaminating somewhere in the process. Another thing we do to help improve our accuracy in our measurements is called a standard addition. If we have a lot of interference from the matrix, that's essentially the solution, all the types of stuff that might be in the sample besides our analyte, we can often spike our samples with a known amount of analyte to make it easier to read. We can think about this kind of like if you're looking in a field for something and you couldn't find it because of all the grass, that's like all the matrix interference in a sample. But then if we took that thing and put it on top of a 10-foot pole, put our object right on top,
then it's much easier to see because it's above all the grass. In the same way, when we spike with additional sample, our signal is a lot easier to see above all the noise. We'll talk more about standard additions and how to do them later. One common difference between procedures and protocols is that protocols often define quality control steps in addition to run specifics, so the actual steps that you do for each sample. For example, we might have an initial calibration value and initial calibration blank, ICV and ICP on this figure. And then we'll regularly compare those to a continuous calibration value and a continuous calibration blank. So we'll have known values of some amount of sample and a zero amount of sample, and we'll constantly be comparing them. By comparing the standards at regular intervals, we ensure that the method is behaving as expected and we can quickly troubleshoot problems.